Thank you to all of the attendees of today's webinar entitled Use of DNA Analysis in Seafood Species Identification. My name is Eddie Deal, and I will be your host for the webinar. Today, I want to walk you through who ACGT Incorporated is, provide background on the seafood fraud issue, define what DNA is, outline our species ID process and the data you will receive, identify who needs our services, provide instructions for seafood sampling and shipping, briefly mention the concepts of traceability and sustainability, describe some additional ACGT Incorporated expertise that adds to our industry leadership position, and finally, make a few concluding remarks. First, I would like to introduce our company, ACGT Incorporated, the provider of the Seafood ID service. ACGT Incorporated has an extraordinary record of DNA analysis service to pharmaceutical, biotech, academic, government, medical, and consumer products scientists. Our staff includes 6 PhD level scientists and 15 master's or bachelor's level scientists. We are compliant with FDA good laboratory practices, which are required for submission of data to the FDA in support of drug or diagnostics applications. How did we become interested in seafood fraud? The problem was noted in the scientific literature, it was noted by the FDA and numerous fishery groups and NGOs, and it was noted in the popular press, most recently by Consumer Reports and the Boston Globe. To address this problem, we created Seafood ID from ACGT Incorporated. Our scientific expertise and global project experience give us unique and robust qualifications to meet the needs of the seafood industry. I want to provide a brief overview of what DNA is, since all of our discussions today are based on the use of DNA analysis technology. DNA is a linear string of paired chemical bases in every living cell. Whether you are a bacterium or a human, you use the same 4 chemical bases: adenine, abbreviated A, cytosine, or C, guanine, or G, and thymine, or T. DNA is used for the storage of information and as a blueprint for metabolism, growth, and reproduction. The DNA sequence is unique to each species and to each individual organism. For example, humans differ from each other by only 0.01% of their DNA sequence, but that is enough to make each of us unique. For our discussion today, the important thing to remember is that the similarities and differences between DNA of different species can be used for identification. Our primary gene target is cytochrome oxidase 1, the so-called barcode gene. Our secondary gene target is cytochrome B. We use the same techniques, technology, and primary gene target as the FDA to identify seafood species. The FDA also published a standard operating procedure in August of 2011, and our standard protocol is as rigorous as the one used by the FDA. This is a flowchart of our Seafood ID species identification procedure. We start by extracting total DNA from a small piece of seafood tissue. Then we amplify the target DNA using the polymerase chain reaction, or PCR. As I said earlier, each cell in every organism has DNA, but the amount of DNA in samples is too minute to analyze directly. So the target sequence needs to be amplified. Once amplified, we can analyze it. Here is a picture of a PCR machine at the top. We clean up the PCR reaction using the Biomech machine. This is above and beyond the technique used by the FDA in their seafood testing SOP. Here is a picture of a Biomech machine in the middle. Once the DNA is amplified, we sequence it using one of our 3730XL DNA analyzers, the same equipment used by the FDA. Once we have that sequence, we compare it to several databases to find a match, and that gives us the species the sample came from. Here is a picture of a 3730XL DNA analyzer at the bottom. This slide is a case study, if you will, on the specific use of seafood identification to distinguish between big eye and yellowfin tuna. The first picture shows the use of electrophoresis to evaluate the quality of PCR amplification products that I referred to earlier. What we want to see here is a large band for each sample. Next, we do the DNA sequencing, and the machine produces the graph shown at the bottom, which is then converted to a series of A's, C's, G's, and T's, the DNA sequence. This DNA sequence is then queried against one or more databases, and in the picture to the right, you see the DNA sequence of the cytochrome oxidase 1 gene from a big eye tuna on top and from yellowfin tuna on the bottom. If you look carefully, you'll see 7 differences, and we will find these and tell you if your sample is big eye or yellowfin tuna. This is an example of a portion of the Seafood ID report you will receive from ACGT Incorporated upon completion of your project. In this case, we use the GenBank and Barcode of Life databases to determine the species of 10 samples. In 2 cases, samples 1 and 4, the species identity was determined to be different from the one provided by the client. Additionally, we provide full species information for each sample and added value to the client. As I mentioned, we use several databases, including the Barcode of Life database, the FDA's recently published sequences, and the GenBank database. All of these are updated routinely. The Barcode of Life database and FDA databases are very well curated and monitored, but there has been some concern about the GenBank database and its accuracy. GenBank contains published, peer-reviewed sequences for thousands of fish species, nearly all commercial fish. For the major fish and many minor commercial fish, multiple sequences are available, collected by different groups of fisheries scientists. That is to say, there are multiple independent data sources available, allowing cross-checking and data verification. Therefore, GenBank contains the peer-reviewed sequence contributions of hundreds of fishery research groups, and this combined resource dwarfs any proprietary database. Advanced techniques for sample analysis. Single species identification is straightforward, but sometimes it is necessary to analyze the species present in mixed and processed products. Often, DNA in highly processed, cooked, or chemically treated products can be highly degraded. In such cases, we need to use alternative methods of DNA analysis. When multiple species are mixed together, one of the technologies we can call upon is next-generation sequencing. Our next-generation sequencing, or NGS, platform is the Illumina Genome Analyzer 2X. It utilized massively parallel sequencing to analyze millions of short DNA sequences at one time. What does that have to do with our Seafood ID service? This technology can be used to analyze all of the PCR products from a mixed seafood sample at once and identify all of the individual species present, including their relative levels in a sample. Next-generation sequencing can also identify new markers for use in traceability and sustainability projects, which I will mention in a few minutes. This is a picture of a team member at our Illumina. Who needs our services? All participants in the seafood industry, including the consumer, have something to gain from DNA analysis. Each client is interested in verification-based value. And as you know, seafood changes hands many times in the supply chain. DNA analysis adds value to the next step by verifying the previous step. Our sampling procedure is simple. Just follow the instructions on our Seafood ID website for sample collection. You can use your own tubes, or we will send you our Seafood ID sampling kit. You can ship samples in tubes at room temperature, but if you prefer, you can ship fillets or whole fish on cold packs or frozen on dry ice. Please note that keeping samples frozen on dry ice is not required, but our clients are welcome to do so if they desire. Unopened cans, jars, or packages are also acceptable. Seafood samples can be raw, cooked, smoked, dried, or canned. If DNA is there, we will get results. I wanted to bring up the issue of traceability. The goal is water-to-fork tracking of seafood. To do this, the barcode gene alone is insufficient, and additional genetic information is required. Therefore, we need reference samples, additional R&D, and alternative techniques. Sustainability is another concept that is gaining popularity. It involves analysis of population genetics and making sure that approved fishing areas or specific fish farms are being used. Again, this often requires reference samples and additional R&D. Whether you are interested in traceability or sustainability, we work closely with our clients. This is a picture of some of our team working with our 3730 XLs and the Illumina all the way on the right. Additional ACGT incorporated expertise. One example of an additional technique that can be employed for traceability and sustainability analysis is called short tandem repeat, or STR, analysis. We recently used STR to complete a rainbow trout population analysis project. The client provided us with a series of gene targets, markers if you will, that we used in this work. We used short tandem repeat analysis to determine the relationship of individual fish within a population. We can also use gene expression analysis to distinguish between closely related populations. This technology can also be used to identify genetically modified organisms, or GMOs. Everything on this slide is custom work, but we are happy to discuss possibilities with our clients. Expertise with a variety of molecular analysis techniques is why we have been a leader in the DNA analysis field for almost 20 years. ACGT Incorporated's expertise and access to a number of different techniques and state-of-the-art equipment are important for the completion of every project, from the simple ones to the very complex ones. This is a picture of a team member extracting DNA from a sample. To conclude, seafood fraud is not going away. DNA analysis is a viable method for surveillance. The market is ready for this technology, and Seafood ID from ACGT Incorporated is ready to help. Thank you for your interest in Seafood ID, and we are looking forward to hearing from you.